Thank you.
Okay, now we will move from packaging to a completely different yet equally vital area, to the gut microbiota. Our next speaker is Dr. Jonathan Gorelick, who brings deep expertise in bioactivity. Please, Jonathan.
Thank you. Thank you, Christopher. Very good to be here. Thank you, organizers. Evening, good morning in the US. It's very refreshing finally to give a lecture in English. When I first came to Israel almost 20 years ago, I had to apologize that my Hebrew was not so good. Now, after here almost 20 years, forgetting a little bit of English. So please bear with me. Okay. So today we're going to talk about introduce you to Israeli urtica. Okay, but it's known as nettle. The story starts actually not in the lab, but out in the forest in different places in Israel, going different trails. Think little shrub which was really not too out of the ordinary, except maybe you want to stay away from it because it'll get stung by it. I think on the research of Professor Avanda from the University of Maryland, where she was looking at the possibly developing Urtica dioica as a functional food to potentially beneficially modulate the microbiota as a treatment for obesity. It's very interesting, how this food, plant, which I thought was, I want to stay away from, could actually be a beneficial functional food. I looked more into it, and I got more involved in understanding metabolic disorder, which obviously in the US has been a problem already for many decades. In Israel, it's already also a big problem. Obesity, diabetes, fatty liver. And what we're more and more realizing is not just about food as far as calories and how it affects the body, but also how it affects the microbiome. It's the population of the bacteria in the gut play a very important role in human health. Definitely when they're negatively affected, that can have implications on health. Definitely metabolic disorders like obesity and diabetes disorders. And so if we can find foods which for the microbiome, so this can be a potential direction for helping us in treating metabolic disorders. So just to see who we have the group. So I'm Jonathan Gorelick from the Eastern Regional R&D Center, auspices of the Israeli Ministry of Science. My partners in Israel, Professor Zachai Madar from Hebrew University from the College of Agriculture and Nutrition. He was involved in animal models. Professor Ronan Chazan, microbiologist from the School of Dentistry of Hebrew University. Unfortunately, they couldn't be here. Maybe they're online with us, I hope. And our American collaborator, Professor Avanda from University of Maryland. Her NIFA research was on, again, like I said, the Urtica dioica. We were going to look at the Israeli Urtica, which is a related species. So Urtica, it's known plants. So, it's eaten. I mean, I've never tasted it, but they say it's like spinach. I wouldn't want to go near it because it has these hairs that are stinging. You have to know how to harvest it from the bottom, because the hairs are only on top of the leaf. But it's been used generations. Traditionally, it's recognized as safe as a food. Used, sold as an extract. Been used to treat a lot of different ailments, reduce inflammation, soothe muscles, for strengthening the lungs. It's used for supporting good digestion. Many uses. It's sold throughout the world, even in Israel. It's not made. It's not grown in Israel. This species is found in the U.S., in Europe, and Asia, not really in Israel. The younger brother, so to speak, is Urtica urens. They call lesser nettle, small nettle, dwarf nettle. It's a smaller annual species found throughout Israel and other places, Asia and Europe, not in the U.S. at all, really. And it's also recognized as safe. It's a food. It's used traditionally also for similar indications, but not really. It's really underutilized. It really was throughout Israel, diverse climates, hasn't really been developed. So the real question that we asked, before we started this project was, can this Israeli nettle be utilized to at least partially ameliorate, all different metabolic dysfunctions, specifically those that are where the microbiome is associated. And that's what this project we did. So basically, we had two main objectives: to characterize the biodiversity, the chemical diversity, biological diversity of the Israeli nettle.
Oui.
And to understand the the therapeutic effects in that animal model obesity. Okay. So basically, we went and collected samples of urtica from throughout Israel, 10 different wild samples, analyzed them chemically, biologically, very, very diverse. I mean, it's not such a surprise. The climate's very different from the Galil down to the Judean hills and by the coast, very different climates and precipitation. And we saw, in fact, very different chemistry. I mean, there were some compounds we found throughout, but very, very wide variations in the flavonoids, the coumarins, and phenols, and other compounds as well. Also, the biological activity also had a lot of variation. We tested in cellular models free fatty acid uptake, cholesterol uptake, in hepatocytes, in adipocytes, model of inflammation, nitric oxide secretion in macrophages. Also, here we saw a range of effects. I apologize, I don't have all the results. I tried to keep to what the organizer told me in terms of numbers. So actually, didn't bring those results. Yes, no, it's we have a tendency as researchers to want to share results. I tried to be good this time, so I apologize that there's not so many actual results, but summarize here. Then we took. So actually, we found that ironically, the one that was most active in all of the biological assays was one actually not far from our laboratory. We harvested largely amounts and we prepared for an experimental diet for the model of obesity. This is together with the professor's time at the university. So it's a high-fat diet, so basically nutritionally induced obesity, 15 weeks on a high-fat diet. Mice develop obesity, metabolic disorder, the different types, pre-diabetes, fatty liver, things that dealing with ourselves. And what we found, so there was a decrease in the weight. Significant, but so practically was the high-fat diet. Here's a normal diet is the negative control. Here you see here the two groups that were given urtica. So we had groups that mice who were given urtica as a plant, ground plant, whole plant, included in the food. And we had extracts as well. We tried to extract and concentrate the bioactive phenolics. So what we're surprised to see actually is that those groups that received the whole plants, so they had a significant decrease in weight. And these actually received the extracts, which we thought would be more active, at least in the cellular models, it was more active, actually increased the weight above the high-fat diet control. Surprising. You see that maybe when you try, we try a lot to learn from nature, but when you try to do better in nature, usually you fail. So actually, the whole plant performed better, and most of the parameters that we tested, the whole plant performed better than the extract. But we saw, improvement in glucose regulation, fasting glucose or glucose tolerance, also in the liver function, in the expression of different inflammatory genes. So reduction is just one example. So this is liver stains of the liver cells. So the red is lipid droplets. You see the normal diet is just very few lipid droplets. High-fat diet is the increase. When they were given the Israeli nettle, significant decrease. Just to compare, we also compared, we had groups of the urtica comparable to the American group. And also here it's also a reduction, although we needed a higher concentration of the plant compared to the Israeli nettle. So that's slight comparable. And then we looked at the microbiome. So I'll just give an overall microbiome. There's a lot of data. We're currently really evaluating the data and confirming it in different models. Fortunately, the microbiologist couldn't be here, so I'll just summarize. But overall, we saw that there was an increase in diversity. So in general, we like diversity, so it's a good thing. Also, in microbiome, it's a good thing. The more species, usually, the more diversity of species, the better. When you have problems, metabolic problems, usually one of the things that happens with dysbiosis is there's a decrease in diversity, much more less beneficial bacteria. And we saw that with the urtica diet, there's an increase in diversity, change in the principal component analysis of the different groups. And not to go into it, but you see that the high-fat diet changed. So here's the normal diet, high-fat diet. The different groups modulated the microbiome community. I wouldn't say restored to normal diet, but it's closer. And if we just look at one bacteria, again, we're studying currently a number of species, but take lactobacillus, which is going to be beneficial in maintaining gut tight junction barriers, membrane integrity, and reducing inflammation, and competition with negative bacteria. So you see again that the Urdic groups had a significant increase in the Lactobacillus. Okay, so basically what we from all this came to the conclusion supporting our initial hypothesis, at least tentatively, that Urdic Israeli nettle, whether it's the bioactive flavonoids or other compounds, or maybe other things, because again, we saw that the whole plant worked more than the extracts, so maybe fibers and lignans and things which necessarily the extract. It was we have support to show that they able to restore the microbiome, increase diversity and beneficial bacterial species. And that translates into benefits, metabolic benefits, reducing inflammation, improved glucose regulation. So where to from here? Of course, many, it's nice in the lab, and how you would translate it into actually changing, public human health, which is one of the things here that we have a collaboration with the industry to try to actually take things from the lab, from the science into society. But there are a few different directions we could take as a functional food as itself, as an ingredient can be put into other products. But of course, there's a lot more work needs to be done. Again, it's not currently cultivated, so we'd have to take Israeli nettle and bring it into cultivation. It grows as a weed, so it shouldn't be too difficult, but if I see that it can be standardized and that the bioactivity is stable, formulation into, for product production, and see if we can scale it up, and obviously make sure that people actually will want to eat it, because we hope. Of course, along the way, we had the FITO, which is our company sponsor. It was very helpful in directing us to make sure that we're going in the right direction, that we have interesting research, but in the end, if there's no market or it's not going to be desirable to the customer, it's not really such a point. So we thank them for all their help. Thank you to all the funders, BARD, NIFA, Israeli Ministry of Science, Hebrew University of Maryland, RLab, Eastern R&D. I didn't put again, I was following the order, so I didn't put it. Of course, we want to thank all the students and all the technicians, everyone is involved. One student is here, doctoral student who helped, and other students who wanted to come. But anyway, so thank you. Let's get on the time,
yeah. Thank you very much, Jonathan. It's a very interesting project. I hope that I wish you could bring some samples to the people here to try it.
I thought of this,
But
yes.
Next time, please try it. And I understand that this project is under kind of maturing to the market in early stages.
We can call up actually
the market,
the industrial partners here, yes.
Okay. Can you come here and tell us about this collaboration, and then we'll answer some questions.
Thank you. My name is Dr. Yoshua Maor. After a PhD at the Hebrew University and a postdoctoral fellowship at Harvard Medical School, I came back to Israel and started in academia. However, I launched my own career with my own lab. We started with drug development, and on the way, we started with dietary supplements, food ingredients, and we started to market those products in over 13 countries. We also have done some work in collaboration with IBBL, Coca-Cola Israel, and now we are actually as an ingredient in the Prigat Celuline. Okay, so we are already on the shelf in Israel and in the world. And when I saw the project, the idea, we saw that invention is one thing, innovation is another one. Invention is when you go and you have a lot of information. However, innovation is when you see your product on the shelf, when people want to get that invention, and then it becomes an innovation. We saw in this project something that could be added as an ingredient, and we can find ways of turning an idea, an invention, into something which is actually going to be everything that a scientist wants. Scientist wants to see its invention turning into something that in the future we can promote and see that the world is getting better through science. So we strongly believe, I don't like to say believe, I say we have evidence that actually this project can become actually very, very soon something like ingredient for foods and going to help people with all the problems that we have with obesity, and it's everything which is connected to obesity. So that's how the interaction between science, academia, and also the industry is becoming quite real in Israel through these kind of collaborations funded by BARD and NIFA and the Israeli Innovation Authority and all that. Thank you very much for your attention.
Thank you very much for your insight. Very interesting. And don't run away. We noticed that you will need the hats too. And Jonathan, this is for you. Please bring more or bring this variety to cultivation. If you need some recommendation on farmers that will help you there, please contact us because we have a list of interesting scientists. Okay, question, please.
No, they need gloves, not hats.
You need gloves, okay.
You have to, from the bottom, there's no hairs. It's only on the upper bit.
Another question to Jonathan before he will leave the stage? Any question from NIFA side? Okay. Thank you very much, Jonathan.
Yes, thank you.
And keep up with the good work.
So now I have a